for that great work. And our next speaker is Srinivasan Sivanandan. Srinivasan is a senior machine learning scientist at NCTRO. He works on developing machine learning methodologies for high content image-based profiling and screening for drug targets. Prior to NCTRO, he received his master's degree from the University of Toronto and was a lead engineer in medical imaging at Samsung. And so without further ado, Srinivasan, you can take it away.
Sure. Yeah, thank you for the introduction, and I'm happy to be part of this awesome workshop. Today I'll be presenting our work on machine learning enabled pooled optical screening in human lung cancer cells. Traditionally, image-based CRISPR screens are performed in arrayed formats, which means the CRISPR knockout of each gene is performed in different experimental compartments, that is, in different wells of a 384-well plate or multiple plates. So in order to perform a 300 gene perturbation screen with about 4 guide RNAs per gene and 3 replicates per guide RNA, we typically need to perform an experiment over 3,600 wells, or like about 10 384-well plates. In 2019, Feldman et al. Introduced a pooled optical screening technology by which we could perturb cells using a pool of guide RNAs targeting different genes, all in a single well. The perturbed cell pool can then be stained and imaged for different readouts we want to observe, such as cell paint, QPC, or antibody markers, etc. To be able to obtain the perturbation identity of each cell, they also introduced sequencing by synthesis chemistry, by which the guide RNA perturbed in each cell can be read out base by base. Here's an example image from a pooled optical screen on A549 cells, where on the left, we have a cell painting image of the cells, and on the right, we have images of the in situ sequencing by synthesis methodology. The image on the right shows base calls observed over multiple cycles of sequencing to construct a barcode per cell that we can then use to obtain the identity of the genetic perturbation that was added into the cell. So each dot here is corresponds to a barcode that's observed over multiple cycles, and each color shows whether it's A or T or GRC, very similar to how genome sequencing is done as well. Pooled screens have reduced batch effects, as we need very fewer wells to observe all the conditions. And it's also simpler to manage the samples, as we have fewer wells, and it's much easier to scale. However, pooled screens are challenging, as it requires complex data processing workflows for segmentation, barcode calling, and barcode assignment to the cells. So let me walk through a workflow for processing pooled optical screens. So first, we have a set of pooled perturbed cells imaged using cell paint in this case. And along with that, we also have the in situ sequencing by synthesis images, which have multiple cycles of these ATGC dots that's observed to get the barcode. First, we use the cell painting images, and then we correct illumination and segment the cells to obtain the single cell boundaries. And in this case, we use cell pose models for segmentation. And then the cells are tiled and masked by their segmentation masks. In parallel, we use the sequencing by synthesis images and to sequence the guide RNA barcodes and to obtain the locations of the guide RNA barcodes in the sequencing space. However, an important point to note here is the sequencing by synthesis images and the actual cell paint images could be taken at multiple different time points at 2 different microscopes, and it could be taken there could be several instrumental and experimental noise that goes in between the acquisitions. So to resolve for that, we compute an affine transformation function to map the coordinates from the sequencing by synthesis space to the cell painting acquisition space. Using this, we can then assign the barcodes to the cells and then the corresponding cell regions, and then map them back into the single cell tiles that we have generated previously. So finally, we have a data set of these single cell tiles along with the labels or like the genetic perturbation that was perturbed in the corresponding single cell type. We can then use these single cell images to learn feature representations that can be useful for understanding biology. In this paper, we demonstrate an improved methodology for in situ barcode sequencing, and also the use of self-supervised methodologies for learning useful feature representations from single cell images. So let's get into the first question, which is, can we improve in situ barcode sequencing using machine learning? Let's look into the existing methodology for in situ sequencing, which was introduced by Philman et al. So this approach involves first registering the sequencing images across multiple cycles, followed by a blob detection method across all the cycles, which are then used to obtain a consensus XY coordinate and the corresponding barcode at that coordinate location. However, there are challenges to using this approach in practice because the field of view images need to be like aligned every time across every cycle, because the field of view images could be misaligned due to experimental and instrumental errors. And so typically to resolve this, the images are aligned during acquisition when they're acquired in the microscope. Second, blob detection methods are noisy and requires manual tuning of parameters. So in this work, we proposed a new methodology for in situ sequencing, where we first train a supervised convolutional neural network model that takes as input a single cycle of the sequencing by synthesis image and produces a map of base call probabilities. We then extract the base calls and the corresponding coordinate locations from each cycle, and then use transformations computed from each of those cycles to the first base call cycle. So we then use the transformed base call locations to compute and stitch a completed barcode at each XY coordinate position and the corresponding gene identity at that location. So in this case, as we know the guide RNA library that was used in our experiment, the base calling method can be easily trained and refined using pseudo labels obtained from filtering base calls that are like actually exactly matching with the guide RNA library that was used in our experiment. Training using augmentation also improved the spot detection accuracy. And this method also does not require manual alignment of field of views across multiple acquisitions. We then evaluated the performance of our sequencing methods based on the percentage of cells that we were able to recover with a valid barcode when using each of these different sequencing methodologies. As shown here, our method increased the percentage of cells that we were able to recover by about 10%, which resulted in larger data sets and sample size per guide RNA, which are much useful for downstream feature representation and analysis tasks. So the next step that I'm going to present is the data from 300 gene CRISPR knockout single cell painting dataset, which I'll be using going forward for learning representations. So let's look at this dataset. This screen had cell paint images, which are like images on the left, where we have different organelles that are stained. And on the right are the corresponding in situ sequencing by synthesis cycles, where the barcodes were sequenced, and we get the corresponding gene identity using the barcodes. So we had 300 gene knockouts in this experiment with 4 guide RNAs per gene, with cell painting as readout, which gave us about 1.5 million cells, all in a single 6-well plate. So using this data set, the next question we ask is, can we use self-supervised representation learning methods to learn meaningful biological representations from single cell images? Self-supervised representation learning approaches have been recently performing really well in natural image representations. In this work, we explored 2 popular methods for self-supervised learning, such as which are SimCLR, which is a simple contrastive learning framework, and DynoVIT, which is a distillation with a no labels model using vision transformer backbone. In addition to vanilla Dyno training scheme, where augmented samples are used for training both the student and teacher networks, we also explored using guide RNA sampled positive pairs and gene sampled positive pairs, where the positive pairs were selected from cells having the same guide RNA or the gene perturbation. After learning self-supervised feature representations using one of these methods discussed above, we normalize the feature embeddings using the controls in each well, perform dimensionality reduction using PCA, filter and compute aggregate embeddings per gene knockout, and then construct pairwise feature similarity graph. We then obtain cluster assignments on this pairwise feature similarity graph using latent community detection to look for clusters of genes which might be having similar phenotypes in the representation space. To evaluate the performance of our latent representations, we compared our gene-gene phenotype similarity network against curated public databases of known gene-gene relationships, such as StringDB and gene ontology databases. We define our evaluation metric as the area under the receiver operating characteristic curve of the overlap of feature similarity matrix with the gene relationships obtained from these public databases. The AUC metric allows us to evaluate the latent representations of genes without a need for having to threshold the embeddings having similar, like embedding similarity values. So each column in the database, in the table here, corresponds to a metric computed, provides the AUC metric computed using the StringDB databases or different Go biological process, biological cellular component, or molecular function databases. And each row corresponds to different self-supervised learning methods and also engineered explicit features that we compared against as our baselines. We observed that dyno models with vision transformers provide marginal gains in performance compared to SimCLR and other explicitly engineered features. From the results above, we observed that dyno vision transformer representation for cell painting can be very useful for reconstruction of gene networks and identification of pathway components in a hypothesis-free way. Here are some example images of cells from some of the clusters identified from our latent space representations. The dyno vision transformer representation from CellPaint provide us a way to observe gene-gene relationships and identify new pathway components in a hypothesis-free way. As shown here, we were able to reconstruct the upstream AKT biosynthesis pathway, AKT signaling process, protein glycosylation, fatty acid biosynthesis, steroid regulatory pathway, mitochondrial inner membrane genes, and more. We also see that even when looking at some of the subclusters within each of these large clusters, we can capture meaningful biological relationships. In summary, pooled optical screening requires complex data processing and robust data processing methods. And we demonstrated how supervised learning methodologies using convolutional neural networks can improve in situ base calling and increase the yield of cells from our experiments. And in addition, we also demonstrated how self-supervised learning can be useful in learning latent representations that can uncover biologically meaningful mechanisms and relationships via phenotypic similarity. As with all cool technologies, this work wouldn't have been possible without my co-authors and everyone at InSitro. We are expanding our teams and are actively hiring at various machine learning positions. If you're interested, come join us at InSitro, where we unleash the power of these technologies on iPSC-derived cells in much larger scales to learn meaningful representations in biology to help cure deadly diseases. Thank you all for your time, and I'm happy to answer any questions here or during the poster session.
Great. Thank you so much, Srinivasan.